Thank you, Susie. That was wonderful. So to lead us off on the research-focused presentations, I'd like to welcome Josh Sommer, the Executive Director of the Foundation. I think probably everybody in this room knows him, but if you're not familiar with Josh's story, he was diagnosed with a skull-base chordoma in 2006 when he was 19 years old. And once he recovered, he sought out a chordoma lab at Duke University, where he was a student at the time and has never looked back. Welcome, Josh.
Thank you, Shannon. Susie, I just got to say, that was awesome. So many things that you touched on really resonated, and I was just thinking, what have I learned? A lot of things, but the thing that jumped to mind was a quote from a book by David Deutsch. He's a kind of a famous physicist in the UK, and he writes about philosophy and epistemology and stuff like that. But anyway, his kind of thesis is that problems are inevitable. It's kind of the human condition. We are living in a world that is far from the ecosystem in which we evolved. And so just the act of being alive and living outside of that narrow band in the tropics where we evolved requires solving a lot of problems. And so that's kind of what we do. That's our niche. So problems are inevitable, but he says that problems with the right knowledge and within the laws of nature are solvable. And I think that is really the big lesson that kind of I've taken away from the last 16 or 17 years, is that with the right knowledge, our problems are solvable. There's no reason to believe that just because we haven't solved chordoma yet, that it's this big challenge, that it's not solvable with the right knowledge and within the laws of nature. And I think that, Susie, your story and the way that you have been able to confront your multiple recurrences and the way that medicine has risen to the occasion to be able to address challenges that maybe even a few years ago would have seemed insurmountable is really a testament to that. And it's not my talk here, but I guess I'm going to go off on a little bit of a tangent. I think for my own experience, having had a recurrence last year, I was always under the impression, I was given the impression early on in my chordoma journey that once you've had a recurrence, that really the options are not good at all. There's not much that could be done. And certainly that there are many patients for whom that is the case. But I think more and more we're seeing that medical technology and medicine in general has advanced to the point where many recurrences are very much addressable. We just heard at the conference these last couple days that surgery and radiation continue to advance very significantly. And I think maybe 10, 15 years ago, the consensus was, well, maybe we've kind of reached a plateau. Maybe we've reached the point where surgery and radiation don't really have much more room for improvement, but they do continue to improve, which is extraordinarily encouraging. Okay, let's see if we can get my talk up there. I see it on the monitor down here. Oh, there we go. Okay. Thank you. Okay, so what am I actually going to talk about? Shannon talked about giving an overview of research advances. Dan Fried is going to talk about some of the extraordinary progress that's happening in the Chordoma Foundation's lab. Dr. Mary Frances Wittekin is going to talk about some of the great research happening at the National Cancer Institute. There's no way to do justice to all that's happening in the field. There is just an absolutely extraordinary amount happening. So what I'm going to try to do is give a very high-level overview and give a gist of where the field has come, and importantly, kind of what's really what are we on the cusp of right now? What's coming next in terms of therapies for chordoma? So I think, as probably all of you know, the Chordoma Foundation's mission really involves three prongs. It's research to advance better treatments, it's healthcare improvement to deliver better care to patients throughout their journey with the disease, and as Shannon has has shared so well patient services to provide a better experience throughout patients' journey with the disease. So I want to focus today on research and again, just provide kind of the high-level gist of what's happening and where we're going. I think I really want to underscore a point that Shannon made, which is that when we talk about research, we're not just talking about better treatments to lengthen life, but better treatments to enable those of us who are dealing with this disease to live better lives, whatever that might look like. And from our surveys of the patient community, we have seen that this disease definitely takes a toll in a lot of different ways. And that's something that we need to keep front of mind as we work towards better treatments. So what are new treatments needed for? What are better treatments needed for? So certainly, for a not insignificant number of us in this community, tumors can no longer be controlled by surgery and radiation. They spread, they metastasize, and currently there are really not great options for individuals in those situations. So we've got to have better treatments for patients with metastatic disease. But in many cases, surgery and radiation are feasible. They can address the tumor, but they leave individuals with significant quality of life challenges. We've got to find ways to either make safer, better alternatives to surgery and radiation, or perhaps obviate the need for surgery and radiation. But eventually, we'd like to get to the point where we're not talking about metastatic disease. We're not talking about local recurrences. We can address the disease kind of once and for all. And clearly, surgery and radiation are going to play a role in this disease for a long time, but can we find ways to augment surgery and radiation, adjuvant therapies that perhaps could be taken around either before surgery or shortly after surgery and radiation to prevent recurrence and ultimately stop progression of the disease. So I think probably as those of you in this room can attest, surgery and radiation is really the primary treatment modality for this disease. And I think that we're all here today is probably a testament to just the extraordinary capabilities that surgery and radiation offer for this disease. I'm in particular after my most recent recurrence, just left in absolute awe of what's possible. But as advanced as surgery and radiation has become, there does reach a point for many patients where surgery and radiation are no longer possible. And the key missing gap right now is an effective systemic therapy, either in those instances where surgery and radiation is no longer possible or in instances where it's quite risky. And so this is not an exhaustive list, but kind of gives you the gist of the current state of play for systemic therapies, and really, I think, highlights a big gap. Here you see a number of drugs that have been tested for chordoma and response rates that are really modest. I think that would maybe be a generous statement. So big need for systemic therapies. So we talk about finding systemic therapies or drugs, and we talk about finding a cure for chordoma, but the more realistic scenario is that there's going to be a need for multiple drugs, multiple treatments. It's not kind of one Hail Mary to find one silver bullet to solve this problem. We know that cancer, and chordoma in particular, is a heterogeneous disease. It varies from individual to individual. It varies even within the same tumor. There's multiple populations of cells, and different tumors within the same person can evolve from different clones and have different kind of genetic underpinnings. So there's going to be a need for personalization, a kind of a toolbox of different drugs that can be applied to the right patient at the right time. We also know that chordoma and cancer generally is a complex disease, and it evolves. And there are very few instances where a single drug is curative or highly effective for cancer. More than likely, there's going to need to be a combination approach. And again, those combinations could vary from patient to patient. And then, of course, cancer evolves. So just because you have a drug that is effective or drugs that are effective for a period of time The tumor is constantly evolving and constantly finding ways to evade or avoid that selective pressure that the drug provides. That's not to say that this is a hopeless or futile problem. There are examples of sequential therapy that can be quite effective. So I think the take-home message is it's not a single cure, it's not a single drug that we're driving towards, it's a toolbox that we're driving towards. And that really influences how we think about the kind of overall research effort. If we knew that there was going to be one drug, one silver bullet to solve this problem, we could put all our resources there. We could say, we think this is going to be the best thing and put the resources there. But the reality is we've got to take multiple shots on goal. And thankfully, there are an extraordinary number of potential shots on goal that could be taken, some of which are relatively low-hanging fruit, and some of which require more research. But that kind of pool of options is continuing to grow in really exciting ways. So where could potential new drugs come from? So there's about 3,000 drugs that are already approved for use for any number of different diseases, and they could be repurposed immediately. These are drugs that are, that reside within the pharmacy and that a doctor could prescribe off-label. 3,000 of them roughly, and if there's one out there that could work for chordoma, we need to know. But in addition to that, there's a slightly wider universe of, at any given time, roughly 7,000 drugs that are in development anywhere in the world for a variety of different diseases. About 1,300 of those are for cancer. And so we'd like to make sure that if some of those drugs could be beneficial for chordoma, that chordoma is kind of at the front of the line to take advantage of them as quickly as possible. And what's really encouraging is that those 7,000 drugs and the 3,000 that exist already, they represent only, they only target roughly about 20% of disease-causing proteins or kind of disease-causing alterations. So what that means is that 80% of the things that can go wrong in your cells that lead to disease or lead to cancer are currently or have historically been undruggable or kind of out of the realm of what medicine can address. But that is really rapidly changing. There's an extraordinary number of new technologies that are enabling previously undruggable targets, previous aspects of cancer biology that were just unaddressable to now be targeted. And we'd like to, if the biology takes us in that direction, ensure that chordoma benefits from those as well. So thankfully, in the last 15 years, there has emerged an incredibly vibrant and energized and active chordoma research community, as we saw I think highlighted really dramatically these last couple days. This is a picture from the research conference. The number of researchers who are dedicated to this disease, representing a tremendous number of specialties, from most basic science and chemistry through translational research and clinical research, is just incredible. And the culture that has evolved and been built in this community is really extraordinary. So many collaborations are forming, so many partnerships, so many, really the atmosphere these last couple of days has really just been electric with so many new ideas and so many new projects and partnerships that have emerged. So lots of activity picking up. So what does this translate to? Well, I guess I mentioned at the outset that our problems are solvable with the right knowledge, and thankfully, knowledge about chordoma is increasing dramatically. So I just went to PubMed and I searched for articles that had to do with the biology of chordoma or translational research or systemic therapies. And here you can see, for a long time, not a lot happening, very little known, very little knowledge being produced about this disease. And then in the last 15 years or so, you start to see a kind of a dramatic uptick. So this is when the Chordoma Foundation started. And I want to just emphasize that I think that there is definitely some causation, but there's also just in general, to some extent, this is a reflection of increases in progress in cancer research generally, and just a tremendous number of new technologies that have enabled us to kind of peer under the hood of cancer in ways that weren't possible before. But I think the take-home message here is that knowledge about chordoma is growing dramatically. And there's as I said, way too many advances to give justice to during this talk. But we've learned about the genetics of the disease. We've supported projects across the globe, thanks to many of you in this room, to subject chordoma to a wide variety of analyses. There's a kind of a group of projects ongoing right now called multi-omics, where you try to understand all the different layers of biology. So the kind of the genes, the proteins that chordoma expresses, the RNA that chordoma expresses, and how all of these things fit together. But I just want to highlight one paper in particular that many of you may have heard me reference last night. Our Uncommon Insight awardees, Dr. Stuart Schreiber and Tanaz Sharifnia, they have recently published really a tour de force to try to, for the first time, map all of chordoma's vulnerabilities. So all of the things that chordoma depends on for survival. And this has been just as kind of thrown open Pandora's box in terms of opportunities to intervene. Now that we know what chordoma is vulnerable to, now that we know what it depends on, we can go look to drugs that are approved that could be repurposed for chordoma, drugs that are in the pipelines of pharmaceutical companies that we could try to go to those companies and say, hey, look, chordoma has a vulnerability that your drug could address. Could we form a partnership? Or in some cases, this has pointed us to vulnerabilities that are not yet druggable, but as I mentioned, extraordinary explosion of new technologies that makes that no longer seem like an intractable problem. Some of these historically undruggable targets that chordoma is dependent upon are within reach given current technologies. And Dan's going to talk more about some of the research that's emerging specifically from this paper and work like it. But I think just to boil it down, the takeaway is because of the knowledge that's being generated, we now have educated hypotheses or ideas about ways to target chordoma. So for a long time, we were kind of feeling our way in the dark, and oncologists would take initiative and try to treat patients with available drugs, but really it was not informed by a deep understanding of the biology of the disease. Now we've got an emerging picture of what makes this disease tick, and that's leading to well-justified therapeutic hypotheses that in recent years we've been able to test in the lab. So here you can see, we've set up, as many of you may have heard in the past, a drug screening program where well-justified therapeutic concepts can be tested in mouse models of chordoma to provide justification for clinical trials. We have tested more than 80 drugs now, which is a far cry from where things were when this program was started. There were only maybe 1 or 2 drugs that have ever been tested in mice. 10 of those have gone on to clinical trials, and here you can see this pipeline of clinical trials continuing to grow. So 11 chordoma clinical trials have emerged from work that the foundation has supported in recent years. I would just note, encouragingly, we heard from about 4 of those clinical trials in these last couple days. And I think that it is safe to say we don't have home runs yet, but we are starting to see some really encouraging signs of activity. And I think maybe an emerging toolbox of therapies that could be available to patients in coming years. I just want to give a note that this can be a lot to keep track of. And so we have, at the foundation, tried to make it as easy as possible to keep track of the therapies that are emerging, that are currently in clinical trials, that have been through clinical trials, that maybe will soon be in clinical trials based upon what we're learning about the biology of the disease. And so if you go to the foundation's website, there is a page that lists all of these drug therapy options that are in clinical trials or that soon will be. But that's only as good as a patient's ability to access those trials. And so to kind of build upon what Shannon mentioned, we recognize that it's not just about making trials available, but making sure that they are relevant to patients and that patients can access them. And so one of the things we're most proud of at the foundation in the past year is the establishment of a clinical trial assistance program so that when clinical trials are available, we don't want travel to be a barrier to participation. Right now, this program is just being piloted for an ongoing clinical trial in Houston, testing a drug called cetuximab, which we heard about yesterday. And I think it's really telling that about 80% of the patients who have participated in this trial have only been able to do so because of the clinical trial assistance program. So I just want to give a huge shout out to Shannon for really your vision in setting this up, and for really the whole Chordoma Foundation team and many in this community who have supported this program. It's, I think, really not only game-changing for those who are the beneficiaries, but also for the research field to know that historically there's been this kind of notion that chordoma trials or rare cancer trials are difficult, if not impossible. But I think we're demonstrating that that is not the case. That's a surmountable problem. So here is kind of a snapshot of the clinical trials that the foundation has supported or that are being planned. I'm not going to go into all of them in detail, but I think suffice it to say there are emerging options. This toolbox continues to grow. But we have to continue stoking research to understand the biology of the disease and to translate those insights into well-justified trial concepts so that we can continue growing this pipeline. So just to kind of put this all together, where have we come? Where are we going? Up until about the early 2000s, there were really no systemic therapy options. No clinical trials had ever been done. Beginning in the early 2000s, there were some kind of bold attempts, including Dr. Stacchiotti, who's here from Milan, to use the initial hints that we had about chordoma's biology to try drugs that were emerging at the time. And I would call this kind of opportunistic repurposing, where there were drugs that were beginning to enter the oncologist toolbox. We had some whiffs or hints of kind of what might be driving chordoma, but it was really just the earliest inklings. And so that list of trials that I showed at the outset with relatively low response rates has been the result of this kind of opportunistic repurposing. And so those are drugs like imatinib and lapatinib and dasatinib. In the last number of years, because of this uptick in knowledge about the disease, we've now kind of entered a new phase where we're taking a much more rational and much more scientifically guided approach to repurposing drugs. And I think that you're going to see in coming years, or not in coming years, in coming months, the results of some of those trials reading out with response rates that are significantly higher than what you saw on that earlier screen. And so these are drugs like afatinib and cetuximab and nivolumab. But I think what's really clear is that the next wave of therapies are going to be even better and even more scientifically well-justified. We're beginning to partner with companies to reposition emerging drugs, and those are things like tazemetostat, FHD 609, and there's a growing list that in particular is coming out of CF Labs. But I think it's also very clear that there's a wave of combination therapies that are coming because of work that's been done across the world and because of work that's coming out of CF Labs. We're on the verge of having biomarker-guided therapies, so knowing which patients are most likely to benefit from which drugs so that you can be spared the potential side effects and toxicity of drugs that are unlikely to work and go to the trials or therapies that are most likely to be beneficial. And then in the coming years, not so far off, there's a wave of brachyury drugs that are on the horizon. We heard from at least half a dozen different groups these last couple days that have made substantial progress towards therapies targeting brachyury. And so I think when we're here next, it's very likely that we are going to be much closer to these drugs. So I think the important point here is that progress needs to continue across the entire R&D continuum, from most basic resource development all the way through clinical research. And at the foundation, we have a real sense of urgency. I think all of us recognize a need for progress to happen at a pace that matches the sense of urgency that we feel, which is not always the case in academic research or in the research ecosystem more generally. I think what we've seen thus far is that we can go faster and we must go faster. And I think that I want to now turn it over to my colleague, our head of target discovery and translational research at the Chordoma Foundation, Dan Fried, who is going to talk about what we're doing at the foundation to really step on the gas and make so much of this research to find better therapies go significantly faster. So, Dan.
Thank you, Josh. And Susie, thank you as well. That was really beautiful. That was an amazing talk, very touching. Okay, and thank you everyone for being here. It's been nice so far over the past almost 24 hours to reconnect with several of you, to meet some of you for the first time who I may have communicated with over email. And I'm looking forward to meeting as many of you as possible throughout the day. So thanks again for being here. And what I'm going to do today is just very briefly kind of more from like a conceptual level, introduce the lab that we launched at the beginning of 2022. I'll give a couple of quick vignettes at the end of the talk, but I'm more going to talk about the concept of the new lab that we launched. And one way to describe it is the missing link in the research ecosystem. And I think just hearing Josh talk about it, another way to perhaps describe it is enabling patients to take greater control of their own destiny. And so that's kind of what is front of mind for us with the new lab. Okay, so when Josh was talking about the research and development ecosystem, traditionally, as I think many of you know, there are two pillars that comprise the R&D ecosystem that kind of are fighting towards the development of new therapies and technologies to improve human health. I've worked in both of these pillars prior to coming to the Chordoma Foundation. So the first is academic research, right, typically taking place in universities, and the second pillar is industry. So this is typically research that occurs in the context of a pharmaceutical company or in a biotech company. And today, I'm going to kind of introduce to you a third pillar of the R&D ecosystem, and that's patient organization-driven research. So a key first example of this patient organization-driven research was the drug screening program, which we launched in 2015. And this was really set up to facilitate the evaluation of therapeutic concepts in mouse models to justify new clinical trials. So this is sort of the last step that is very critical in order to generate a data package that can justify the advancement of a therapy into clinical trials. And that was a major step for us because it's led to the initiation of several new clinical trials for chordoma. So Josh mentioned the cetuximab study that is ongoing currently at MD Anderson down in Houston. Another example is the palbociclib study that's ongoing in Europe. And so the drug screening program was a great first step for us. When I joined the foundation at the end of 2020 and throughout my first year here, I realized that there were still several key efficiency gaps that were delaying or even in some cases preventing the continued evaluation of therapeutic concepts. And so this is kind of what motivated the launch of what we call CF Labs at the very beginning of 2022. So Josh showed this slide in his presentation, and really we don't need to rehash it, but the key is that there are several potential paths to better systemic therapies for chordoma. There's drug repurposing of approved drugs. You can reposition drugs that are in clinical trials. You can develop new drugs. But the key here is that regardless of the path taken, they each must progress along several stages of preclinical development, right? And in order to improve care on a timescale that matters to current patients, that matters to many of you sitting out here in the audience today or joining virtually, is that seamless progress is needed along each step of this path in order to achieve care improvement in the clinical treatment paradigm. And for various reasons, progress often is not ideally efficient. And there's a number of reasons for this. So for example, in academic research, there's this huge pressure so career success requires the development of very deep niche expertise in a specific technology or a specific biological pathway, for example. And because of this push towards extreme specialization, academic labs often really aren't built to run the gamut from target discovery to translational research and to clinical research. It really requires collaboration and rapid data sharing in order to achieve this. But there's the path to career success in academia sometimes creates barriers to collaboration and rapid data sharing. And so some potential therapeutic concepts end up stalling out, right? And so this is particularly true for kind of a smaller rare disease research community. So it kind of creates this expertise siloing in some cases. And so we really need to kind of grease the skids. We need to find ways to advance therapeutic concepts more efficiently. So on the other hand, on the industry research side, pharmaceutical companies are structured specifically to advance new discoveries all the way to clinical trials. But there's this extremely high financial risk that's required to achieve a new drug approval. And so as a result, companies typically develop new drugs in the cancer type with the largest possible market. And so this means that oftentimes, drugs are not initially developed for chordoma, resulting in a time lag, right? And so even in cases where there is interest in chordoma, companies usually lack the chordoma-specific expertise or research resources, including assays and experimental protocols that are required to efficiently generate a well-justified data package that can motivate drug development in chordoma. So, all of that being said, I want to be very clear about something here. Researchers and companies pursue cancer research for the right reasons, right? We're all in it in order to make a difference, in order to change the way that chordoma patients are treated and to change the outlook. But, at the end of the day, oftentimes, researchers are accountable to department chairs or investors and not patients. And this can create a situation where, despite the best of intentions, the pace of progress is dictated by the pressure to publish or the motivation of companies to invest in chordoma, and that's not always aligned with the sense of urgency that's felt by chordoma patients, right? And so I think we've been somewhat blinded to this sometimes misalignment of priorities due to the fact that academic and pharmaceutical research have made so many landmark advances that have resulted in huge benefits to human health. I don't think anybody can argue with that. But the fact is that we want progress, and we really need the pace of progress to match or at least approach the sense of urgency that's felt by chordoma patients. So we kind of came to realize that a patient-centric approach represents the missing link in the research ecosystem. And it's not meant to replace the way that research is currently done or even disrupt the way that research is currently done, but rather to augment it by contributing a number of advantageous characteristics to the research ecosystem that can counteract the current limitations that are kind of placed on the pace of progress. So with that in mind, we launched Chordoma Foundation Labs at the beginning of 2022. We're in this shared lab space in Durham, North Carolina, called BioLabs. And over the first almost year and a half, we've hired two excellent research scientists. Oh, I'm sorry about that. We've hired two excellent research scientists, Nindo and Lee, who are here today. They have helped us establish a scalable infrastructure that will allow us to grow seamlessly. We've launched the first several projects in the lab. And we've presented two posters recently this year at AACR, the American Association of Cancer Research Conference, which is the largest cancer research conference in the world. And we've generated data so far that has helped motivate the initiation or planning of 6 clinical trials for chordoma patients. And now in our second year, to meet exponentially growing demand, we're applying for several federal grants, and we're continuing to grow our team and try to add to our capabilities. Okay, and already we've seen a glimpse of how this new lab can work with academic researchers in the community and work with companies in the research community to catalyze advances in chordoma research. And so I'll give you just a couple of quick examples of how we're working along each one of these paths: drug repurposing, repositioning investigational drugs, and developing new drugs to try to catalyze progress in chordoma research. Okay, so on the drug repurposing side, I think many of you are aware of EGFR and EGFR inhibitors. So it's been long recognized, probably for over 10 years, that EGFR is an attractive therapeutic target in chordoma. And this has motivated several phase 2 drug repurposing trials with afatinib and cetuximab, for example. But kind of from a scientific perspective, the EGFR gene is not mutated in chordoma like it is in lung cancer, for example, or some gliomas. So what determines the response to EGFR inhibitors is unknown. So how do we find the chordoma patients that are most likely to respond to EGFR inhibitors so that we can ensure that they are given an EGFR inhibitor? And then how do we find the chordoma patients that are less likely to respond to EGFR inhibitors so that we can spare them the side effects and unnecessary side effects and lost time of an ineffective treatment? So this is a key question that has remained unanswered for some time now, so we decided to tackle it in the lab. And I think a good example of this question is shown here. So this is some data in chordoma mouse models. So I'm showing you kind of chordoma mouse model number 1 here. If we treat mice with afatinib, we see that the tumors completely stop growing. So this is how the tumors grow in untreated mice. Afatinib really suppresses the growth of chordoma tumors in model number 1 over here on the left. In chordoma mouse tumor model number 2, untreated mice, the tumors grow, and if we give these mice afatinib, the tumors still grow, right? So how do we find these patients and separate them from these patients? That's the key question. So we're studying this in the lab right now, and we don't need to dive too deep into the details of this graph, but basically what we've done is we've screened a large panel of chordoma cell lines, and we've identified 2 different groups of chordomas in the lab. So there's a blue group here that is very sensitive to afatinib, and then there's a red group here that is insensitive or resistant to afatinib. And then we profiled our models using a genomic sequencing and transcriptomic sequencing to understand which genes are mutated and which genes are turned on and turned off. And we worked with a group of researchers led by Kurt Bachman and J&J to understand which genes are upregulated in resistant cells or in sensitive cells. And what we found was that in resistant cells, there was a set of genes that are involved in antiviral inflammatory signaling that are upregulated. And so this is very interesting to us right now. It's still kind of early days, and it's just a hypothesis, but it's a hypothesis that we're now testing in the lab. And it might mean that certain patients that are resistant to EGFR inhibitors could potentially benefit from immunotherapy, for example. So we're trying to rapidly advance this hypothesis and test it, and the research is still in progress here. So I should note that it does need further validation, of course, before guiding patient care, but it's something that we're working on in the lab. Okay, as far as drug repositioning goes, to give you an example of some of the things that we're thinking about in this area, a chordoma medical oncologist that doesn't have his own research lab but had a great idea approached us and proposed an interesting hypothesis. And his question was, do some chordomas have defects in how they copy their genome as those tumors grow? And this is a phenomenon called replication stress. And it just so happens that there's a new class of drugs that target replication stress. They're called ATR inhibitors, and they're now in clinical trials. And so we've been collaboratively testing this hypothesis in our lab, and what we have found is that, again, just in some examples here, in chordoma mouse models, if we give them an ATR inhibitor, it, in some models at least, substantially slows the growth of those tumors. There's another drug that can attack replication stress. It's an older chemotherapy drug, actually. This is kind of more of a drug repurposing play that can even shrink tumors in some cases. And then we asked, well, what happens if we combine an ATR inhibitor with gemcitabine? In chordoma cell lines, you see the red curve here is gemcitabine, and then if we combine it with an ATR inhibitor, we see that curve shifts to the left, which is exactly what we were looking for, and that means that these 2 drugs are working together synergistically to inhibit the growth of chordoma cells. So now we're kind of further testing that hypothesis in the lab, and we're testing other drug combinations, and we're in discussions with multiple biotech and pharma companies that are developing these drugs about paths to clinical trials in chordoma should the data continue to look promising. Again, finally, on the drug development side of things, One of the major efforts that is getting up to speed in the lab right now is trying to enable and incentivize drug development against brachyury, which I think many of you know is a hallmark of chordoma and is one of its greatest vulnerabilities. And so what Lee in the lab is working on now is developing an assay pipeline. So how can we design a series of brachyury-specific and chordoma-specific experiments that we can provide to companies or researchers that are interested in understanding whether their technology or their ideas might be effective in targeting brachyury, so that we can kind of facilitate rapid go, no-go decisions for these companies with limited upfront investment to lower the entry barrier in terms of considering brachyury as a target or a program within their drug development pipeline. So that's another thing that we're working on. And so along with a wonderful and highly motivated group of colleagues and advisors, I think we now have the ability to go from target discovery to well-justified clinical trial concept in a matter of in some cases, months, which is fantastic. It's kind of unreal. And it gets us closer to advancing chordoma science at a pace that matches the sense of urgency felt by patients. So that's what we're trying to do. We're trying to reduce these timelines here by working with academic researchers, working with drug companies, and with this patient-centric approach. I'm just really excited for the road ahead. So thank you all for being here. And I don't know, Shannon, if we're doing Q&A or if I should just go ahead and introduce. Okay, I'll take questions at the Ask the Experts session, says the boss. So, okay. Next, I am pleased to introduce Drs. Linny John and Mary Frances Wedekind, and they're from the National Cancer Institute. They're part of a team called the MyPart Network, which focuses on rare tumors, and along with the rest of their team, they are quickly becoming chordoma experts, some of the leading experts in the field, particularly in the realm of pediatric chordoma. Their dedication and commitment to this disease and to the chordoma community are very much appreciated, and we're glad that they're joining us today to talk with us about their natural history study of rare solid tumors, which is being conducted at the NCI in Bethesda. So, Mary Frances and Linny, thank you.
Hi, everyone. Thank you so much to the Chordoma Foundation for inviting us to be here, and especially Shannon for letting us give this talk today. I am Mary Frances Wedekind. I am a pediatric oncologist and research assistant in the pediatric oncology branch at the National Cancer Institute at NIH.
And I'll just quickly introduce myself. My name is Dr. Linny John. Just like Dr. Wedekind, I'm also a pediatric oncologist at the NIH, and both of us have been there since 2019. So for the last 4 years, we're really focused on chordoma and learning more about this disease.
So I would like to just start by saying thank you to all of you for being here today and for the participants that are already a part of our protocol. We would not be where we are today without you. And for the rest of everyone that hasn't heard about our protocol, really the talk today is about what our natural history study is, what the MyPart program is, why we care so much about having engagement with the patients and advocacy organizations like the Chordoma Foundation, and then also just to give a brief overview about the chordoma patients that are currently on our protocol. So starting off, just looking at rare tumors. As many of you already have a rare tumor, you know that there are many challenges. For us at the NIH, we do have the definition of a rare tumor being less than 150 cases per million per year. This means that there are about 27% of all cancer diagnoses a year that are considered a rare tumor. There are many challenges, including having a difficulty of establishing a diagnosis because of the lack of familiarity. There's an incomplete understanding of the biology and the natural history, and there's no standard of care for many of these tumors, and it's very difficult to conduct some of those very important clinical trials. So with this in mind, and knowing that there is an overall lack of knowledge, resources, and community for so many rare tumors, the My Pediatric and Adult Rare Tumor Network was formed utilizing Cancer Moonshot funding. The goal of this network is to really engage all of those involved in rare tumors, from patients and advocates to healthcare providers, lab researchers, to really work together to change the lives of young people with cancer. Through multi-institutional collaborations, we were able to form this natural history protocol. We hold workshops and symposium. We host multi-day clinics, which many of you here have been a part of. And we do all of this through great collaboration with the ultimate goal to have clinical trials for our patients. So looking a little bit into our natural history study, what this means is this protocol is not a treatment protocol. So we're not giving a medicine. What we want to do is overall have a better understanding of multiple different rare tumors, with chordoma being one of our special interests. So what this means is that in a very standardized way, through multiple years, we have patients come onto the protocol. We look at their medical records from the past and then follow them through time, through the future, continuing to collect all data that we can. We also have patients and participants fill out questionnaires. So this is the medical forms, our family history forms. This is also patient-reported outcomes, as Shannon and Josh were saying earlier, how important that is for us to understand what your experience is. This will help us as researchers know how to look at clinical trials and what's effective and what's not in the future. We also, in collecting those medical records, as I was mentioning, we ask very specific questions, and we look through all of those records to pull out as much data as we can so that we can have a better understanding moving forward about chordoma and multiple other rare tumors. So one of the criteria, really the only criteria for a natural history study, is that a patient has a rare tumor. They can be a child, adolescent, young adult, all the way up to adult. It also includes their biological relatives. Participation can happen at the NIH or from the comforts of your own home. We also are able to provide treatment recommendations, and this includes clinical trials not only at the NIH, but across the United States and internationally if needed. So once you are a willing participant and want to enroll on the protocol, really what happens is that we help you collect your medical records. You then sign the consent. We get pieces of your tissue and we look at them under the microscope with our pathologist and then look at the genes of your tumor. We have a weekly molecular tumor board, and what that means is that from the pathology side and from the genes of your tumor side, we meet with all the specialists at NIH and talk about this. From there, we give you those results back with the help of our genetic counselor and provide those treatment recommendations that could possibly come out of that. We have the opportunity to also enroll in our chordoma sub-protocol, where we ask some more specific questions. In a future slide, I will show you some of those more specific questions that we do ask. You are able, again, to come to the NIH if that is of interest to you, and we are able to perform further clinical and research analysis. So looking a little bit more deeply into some of the things that we do ask of our participants, most of this can be all done from home. And again, you do not ever have to come to the NIH, even though we do, especially for our chordoma participants, encourage that. But we collect medical records. We look at things within your medical records as to what symptoms did you have when you presented? What treatments have you received? How long have you gone in between one treatment to the next? What does your imaging look like? What does your tissue look like? And very specific questions about your tissue. With the medical and family history questionnaires, is there something with your medical past? Is there something with your family history that has led you to getting chordoma? Looking at our patient-reported outcomes, we look at numerous different measures, looking at pain, depression, fatigue, and then we also, with the chordoma-specific protocol, ask very specific questions, which again I'll show in a future slide. If you do decide to come to the NIH, we do a few more things so you're able to meet myself, Dr. John, we have a nurse practitioner, we have our genetic counselor, and we have our neuropsychologist. You're also able to meet multiple other members of the team. We take blood work. We can do biopsies or imaging if clinically necessary. And then we also talk to you about any treatments and any recommendations that we have. We are also able to do just a few more special tests with our neuropsychologist if clinically indicated. So looking at our biospecimens, and biospecimens is your blood and your tissue and your saliva. So as you can see, those are the three things that we collect from your tumor tissue. As I mentioned, we take that tumor tissue, change it into the DNA and RNA, and look for possible mutations, and then look at multiple other research analysis. From saliva, we do send these kits to your home, and then you send it back in a prepaid envelope. We do change that into the DNA, and then look at that through clinical germline, so what's all across your body in changes, and then also other research analysis. And then for the blood samples, we're looking at the immune cells and just looking at all of those that are circulating through your body, and also cytokine levels. All of these assessments that occur on our protocol when you come to the NIH are never billed to you or your insurance, and we also do have funding to support travel and lodging. So this is a more simple view of how everything goes. We do have some pamphlets in the folders, and this is some of just the handouts that we give our patients. But as you can see, we, from the very beginning, you fill out online some of the questionnaires. From your own home, we send you the saliva, you send it back to us. We ask for the tissue and the medical records. From there, we change that saliva and tumor tissue into those deep components of the genes. We analyze that and then let you know those recommendations and those results, and if there are any treatment recommendations. And then at the clinical center, we continue to do further research and hopefully bring you so that we can also meet you in person and do some further testing. So to look overall at our entire natural history study, we opened the protocol in January of 2019, and so far, as of a few days ago, we have enrolled 551 participants of all age ranges. This includes 6 different continents with 22 countries outside of the US, and most states within the US included. This slide is a little bit busy, but the most important thing is that the #1 diagnosis that is included in our protocol is chordoma, and we did just enroll our 100th participant just last week, and so it's very exciting. And without the Chordoma Foundation, we would have not have gotten here, and without all of your participation as well. So really excited to have that as our top participation. The rest of the list is just other rare tumors that are included. So looking at neuroendocrine neoplasms, FSH-deficient GIST, adrenocortical carcinoma, and medullary thyroid carcinoma. And these are just different rare tumors that occur across the body. So another important aspect of our program is returning results to you and to the patients. This is so important. Like most institutions, we do have a patient portal. We do, as I have mentioned, communicate the treatment recommendations. We do have that weekly tumor board, and we like to talk to you and to also your physician anytime that we are talking about those treatment recommendations. We also, through our communications and advocacy reach out, we do want to provide summaries of our enrollment through our newsletter and social media, and also provide lay summaries of our publications that do come out. So now there are multiple reasons as to why having a partnership with you and our advocates is so important. As Dan was talking about, the rigors of research can really bog people down. But when we have interactions with participants and advocates, we really remember what we're doing this for. And the patients and the faces behind the research are what continue to drive us every single day. The other aspects are that you provide a lot of knowledge. You know, you are the ones that are experiencing this, and we need to listen to your voice and learn from you. You have a diverse knowledge of this disease that, unless some of us have a chordoma, that we will never experience. Other benefits is that it really stimulates discussion. You know, again, from your viewpoint, we can have more in-depth discussions about what we should be looking at, what challenges you're facing, and how we can help you from the research standpoint. This also just spurs the innovation that comes out of the research that we're doing. So as the Chordoma Foundation has been moving into opening a lab, this is where so much of that innovation is coming from, and being able to partner with advocates and patients, you know, the future is very bright if we all work together. And so, you know, why does my part want to do it and do it right? And that's because patient engagement is so much more than just having more patients on clinical trials. Not that that's not important, but as I've kind of talked about, it's so much more than just having more people participate. With a successful partnership, we can have a two-way flow. We provide you with information, and you provide us with such valuable information. It also enhances your participation in the research and keeps you engaged, as well as keeping us engaged. And for the patients, you guys want results, and we know you want results, and you want results for so many different things. You want not only your own results, but to know the results of the bigger whole. And then having this relationship really is impactful for everyone that is involved: the patients, advocates, organizations, and all of the researchers, clinical and lab researchers included. So currently, we have 28 advocate partners, including the Chordoma Foundation, and we are very grateful and thankful for all of these partnerships, and continue to increase them as we are able to. Another important aspect of our program are our rare tumor clinics. A few of you in the audience have been a participant and participants in past or our most recent. So these were all started with one of the other rare tumors, the wild-type GIST clinic. But these clinics, we bring about 10 to 15 patients to the NIH. We also bring disease experts from across the country. So it's not just us here at NIH, but experts from across the country, so that we can really take a deep dive and really help the patients that are coming to the clinic. So in this 3-day event, we have detailed clinical analysis, we do our patient-reported outcomes, we have focus groups, and then as patients, you get to meet with the experts. And we do ensure that all of the experts are there. You know, with chordoma, there are multiple different aspects. So surgeons, radiation oncology, multiple different surgeons, medical oncologists, pediatric oncologist, radiologist. We ensure that all of those physicians and experts are there so that we can have a very meaningful conversation. The other benefit is that you as the patient get to meet others, you know, particularly for our pediatric and our young adult population. They may have never met anyone that has a chordoma. And so to meet someone else their same age that has a chordoma and have that connection, we have been told by the patients is that's one of the very impactful things about these clinics. Participation can happen in person at the NIH, and we do have an option for remote participation as well. So going briefly just through, you know, how we started with chordoma and specifically pediatric chordoma. Within NIH, there was a glimmer of research with chordoma. A workshop was formed, and that's where our partnership with the Chordoma Foundation started. From their help, we were able to make our chordoma sub-protocol and then start our inaugural pediatric chordoma clinic. From there, we were able to meet multiple experts and start many collaborations, which include a multi-institutional virtual tumor board that is held at MGH with Dr. McDonald. And then we were able to make pediatric chordoma working groups to really dive into some of the very specific questions that we needed to be asking with our study. And again, the end goal is to have these treatment trials for our patients. So as I mentioned a few slides ago, with our sub-protocol, these are our patient-reported outcomes. So on the left-hand side in the blue, these are on our, you know, bigger protocol. When we look into our chordoma-specific protocol, we ask very specific questions, and these are based on the location of your tumor because that's very important when we think about what symptoms you're having. And so, as I mentioned, these were developed with the help of the Chordoma Foundation and many of the experts in the field.
Sorry, I'm shorter than Dr. Wedekind is, so I had to move this down. So I just wanted to take the last portion of this talk to tell you a little bit about the patients that are enrolled so far onto our chordoma cohort of our natural history study, and then just take a second or two to talk about one of our current ongoing clinical trials that we're conducting at the NCI. So first and foremost, this graph that you see here is our cumulative enrollment of patients onto the natural history study who have chordoma. When our study started in 2019, we were slowly trying to get the word out. As Dr. Wedekind mentioned, the Chordoma Foundation was very beneficial in helping us do that. I think many of you have probably received some of those email blasts that they've sent out. But since that time, as Dr. Wedekind mentioned, we've slowly started to get that enrollment up and up. And last week, we did enroll our 100th patient, which is an exciting milestone for us. And I know some of you here in the room are on our study, some of you listening on the live stream are on our study as well, so thank you so much for your participation. And just to briefly just show how diverse that group of patients with chordoma are, you can see on the pie chart all the way to the left, this shows the age of the patients with chordoma at the time of diagnosis. And we have patients who were as young as 1 year when they were diagnosed. Our oldest patient was diagnosed at the age of 75. You may also know that pediatric chordoma is fairly rare in our chordoma population. We usually generally see pediatric chordoma in only 5 to 10% of cases. However, with our interest in pediatric oncology, we've taken on to try to get more pediatric chordoma patients onto our studies. So 31% of our patients are pediatric chordoma, which we define as less than 18 years. We're also interested in our AYA population. That's our adolescent and young adult population. We define that as 18 to 39 years, and we actually have 26% of our cohort falling into that AYA population, which is great. And while most most of our patients are from here in the US. We do have about 25% of our patients that do reside internationally. They either heard about our study through their physicians or through the Chordoma Foundation, or honestly, even just by Googling and hearing about our study that way. So it's great that we have a pretty large, diverse group in terms of the patients that we have. In addition, because we have such a large cohort of pediatric patients, one thing we know is that in pediatric chordoma, the most common location is in that clival area, so the base of the skull. And because we have that large population of pediatric patients, that is the most common area that we do find our patients to have tumors. But in the general population, we actually see tumors more commonly in our sacrococcygeal area, so right at the bottom of the spine, and that is the second most common location that we see. Also, just in the general population, the most common subtype of chordoma are our conventional chordomas, and that's represented in our cohort as well. About 90% of our patients have conventional chordomas, but we do have patients as well that have poorly differentiated chordoma and a patient with dedifferentiated chordoma. And these rare subtypes of chordoma are of special interest to us as pediatric oncologists because we generally see them more often in the pediatric population. And unlike conventional chordomas, we still don't have consensus guidelines on how to treat these rare subtypes. And while most of our patients did not have metastases at the time of diagnosis, metastases meaning spread of disease to other areas of the body, 25% of our patients did have metastases either at the time of diagnosis or later on in their disease course. So as Dr. Wedekind mentioned, and it's important to state again, our natural history study is simply an observational study. We do not provide treatment as part of the study. However, of course, we do let patients continue on with the treatment that they're already receiving at their primary institution. But it's important for us to collect that information to really learn what's working, what's not working in regards to treatment. So this larger graph here, this bar graph, is just a summary of the treatments that our patients on the chordoma cohort have received. And you'll see from that first bar, many of the patients have received surgery. And that makes sense because we do know that surgery is part of the consensus guidelines for treatment of chordoma. And most patients did receive some sort of upfront surgery. You can see from the second bar that most patients also did get radiation. In this bar specifically, we're talking about adjuvant radiation. So this is radiation that was given after surgery to take care of any tumor that's left behind. One of the questions that we really want to look into further and find an answer to is which patients need radiation and which patients don't. So having a study like this helps us look into that and see when did radiation help? When was radiation potentially not needed? Does the size of the tumor matter? Does the extent of the surgery matter? And so this data will help with that. We did have a few patients who also received chemotherapy. One thing we've learned is chemotherapy generally is resistant to our most common type of chordoma, known as conventional chordoma. But for one of those rare subtypes of chordoma, known as poorly differentiated chordoma, we have learned that that tumor is more chemosensitive. So the patients that got chemotherapy were the ones with that rare subtype. And then we have a few patients who received targeted therapy. So drugs like immunotherapies and other therapies that target specific molecules. Generally, this was reserved for patients who had advanced disease, so progressive disease or recurrent disease. Those were the ones that received that therapy. Since the time of enrollment, unfortunately, we did have 5 patients who did pass away after the time of enrollment. But for the other patients, we do have a fairly split population of patients who continue to have disease and those who don't have disease. And once again, that's really important for us to look at and measure to see what treatments have been working and what treatments haven't been working. As Dr. Wedekind mentioned, we also do genetic testing on all of our patients with panels that are done at our lab of pathology. Sometimes we're unable to obtain tissue from our patients for one reason or another. Other times, unfortunately, just given the bony nature of chordoma, it's hard to extract the DNA that we need. But for the other patients, half the time we were able to find mutations, and other times we were not able to find any mutations when looking at their chordoma tissue. And last, I just wanted to mention a study that's currently ongoing at the NCI. This is a trial looking at two medications, tiragolumab and atezolizumab. So try saying that 10 times fast. But with this trial, specifically, we're looking at tumors with SMARCB1 or INI1 loss. So in the realm of chordoma, we're specifically talking about patients with poorly differentiated chordoma. Unfortunately, patients with conventional chordoma or dedifferentiated chordoma are not eligible for this trial. The other eligibility criteria is that you have to be between age 1 to 39 years old. We do have an exception. If you are part of our NCI cohort, we can expand that up to ages greater than 39 years. But if you are interested in learning more about that study, we can certainly answer any questions you have. So in summary, just as a reminder, with our MyPart team, we're really here to engage researchers and advocacy partners to really pave the way to more research in rare tumors, including chordoma. But as Dr. Wedekind mentioned, you guys here, you guys listening on the live stream, the patients, the family members, all of you affected by chordoma are the ones that are most pivotal in this research. We rely on you guys to tell us what's important to look at and really help us pave the way into making new clinical trials. So with that, I want to thank our MyPart leadership, the Chordoma Foundation, also all of our other collaborators and advocacy partners. It all takes a village, but most importantly, all of you, the patients and the family members. So if you'd like to learn more about our natural history study or any of the other initiatives that we're doing with MyPart, you can go to our website, cancer.gov/MyPart. It has a lot of information there. It also has our general email address if you would like to contact us. I am not Twitter savvy, but for those that are, we do have a Twitter handle as well, so please follow us. I think that's what you do with Twitter, you follow. So please follow us. And then as Dr. Wedekind mentioned, in your folders, you do have some more information about our natural history study, a pictogram, frequently asked questions. We're also going to be in the back during the break, so if you have any questions for us, please come out, or if you just want to say hi, please come and say hi to us. And if you feel like you're ready to enroll onto the study, the second page of the packet is a ROI, or Release of Information form. What that simply does is if you fill it out and sign it, it gives us consent just to reach out to your treating institution to, in a confidential manner, obtain your medical records for review. And once we do, we'll reach out to you, go into more information about the study, and obtain your official consent to join the study. So if you feel comfortable, you can absolutely fill that out right now, give it back to us, but otherwise, please just take some time, think about it, and ask us any questions that you have. Thank you.
All right, so it's time for a break. We're going to have about 20 minutes, so if we can be back here at 10:10 so we can get started with the Ask the Experts panel. This is the form that Dr. John was just talking about. Doesn't commit you to anything, it just makes sure that they can follow up with you. So if you want to do that, take that out of your folder so you can go and talk with them. And the natural history study data that they just mentioned will help us with clinical trials in the future. It'll help identify drugs that may be working to inspire new trials. And so it's just really critical, important data. So thank you to everyone that's participated and is considering participating. And then if you have questions for the panel, you can put them on this card and hand this to me during the break. See you in 20 minutes. Thanks.